Great, thank you. Thanks for coming today. New venue, so you've all passed the first test and found it, so that's fantastic. It's wonderful to see you all here, and we really look forward to these events. So it's really appreciate you being here. So I'd like to formally welcome you to today's event for the eDNA for Global Environment Studies, which is an exciting BHP and Curtin University initiative that centres on environmental DNA, or eDNA, and its use as a biomonitoring tool for biodiversity. So today is the 5th program update that we've had, the first for the second extended program or expanded program that was announced last year. So a new set of projects to talk to you about today. So I'm Shane Herbert, I'm the program manager for the initiative and that the team will be presenting on today, and I'll be your MC. So before we get started, I'd just like to make an acknowledgement. Curtin University acknowledges all First Nations of this place we call Australia and recognises the many nations who have looked after our country for more than 60,000 years. We are honoured and grateful for the privilege to maintain campuses operating in Boorloo, Perth, and Kalgoorlie in Australia. We pay our respects to elders past and present as custodians and owners of these lands. We recognise their deep knowledge and their cultural, spiritual and educational practices and aspire to learn and teach in partnership with them. Curtin also acknowledges the First Nations peoples connected with our global campuses. We are committed to working in partnership with all custodians and owners to strengthen and embed First Nations voices and perspectives in our decision-making now and into the future. Okay, I'd also like to address a few housekeeping matters. So in the event of an emergency, please exit through the door that you entered just down the hallway a little bit. There's a stairwell just past the entrance to the lifts there. Take it down one level to St George's Terrace level. And in the case of fire, of course, please don't take an elevator. The muster point is down south down William Street. Where are we? Oops, down that way. And it's on the corner of Mounts Bay Road and William Street there. Toilets are located just outside, so the men's are on this side and the ladies are on the other side. Can also ask you to take a moment, just check your mobile phones and they're set to silent. We've organised refreshments at the conclusion of the presentations. However, in the meantime, there's a number of options in this new facility. Carsten's breakout area. We've got water, coffee, and even lollies, an award-winning lolly bar apparently for an energy boost if you're up for it.
It only has 4 different types of lollies.
already been thoroughly scoped out. Excellent. So please help yourself. So refreshments are going to be downstairs at Carsten's from 4 o'clock, which is located downstairs, and we'll show you at the end of the presentations where it is. We'll have lots of people guiding you the way. I really encourage you to come along. That's often a very valuable part of the day. This presentation is also being broadcast by Teams. We've muted the lines for our online attendees, and everyone's still hearing us okay, Sam? So, good. So if you've got any questions, please pop them in the chat, and my colleague Sam here will pick them up and put them to the speaker where we have time. We don't have a lot of time for questions, but we did want to include them. We're also going to try and capture today's proceedings in a recording and make it available afterwards. There's a lot of people that couldn't make it today. If anyone has any privacy concerns, then please come and let me know, or someone from the Curtin team will do their best to block you out. Okay, thanks for your understanding. We'll get into the presentation. So with that, the presentation, we've got 9 projects to cover today. So we've grouped them into ecology focus, and I'm going to introduce them in that way. So we've got a couple of minutes after each talk to ask you questions, but we'll have to move on. So as you can see up there, the first theme is wetlands conservation. And I'll introduce the speakers shortly. The second one is marine conservation. Third is terrestrial biomonitoring, and the last is new technologies. So first up, we've got 3 speakers. So we've got Dr. Angus Lawrie presenting Living Waters of Western Australia. The second one, Dr. Nicole White, talking on eDNA and moving beyond the barcode and subterranean fauna detection. The third is Dr. Mattia Sacco, who's recording today, coming in from Paris, I think he is. Hypersaline and groundwater conservation through eDNA approaches. So I'd like to ask Angus to come to the stand, please.
Maybe I can talk.
Yep, that's on my next slide that I didn't cover off on. But yes, I'll introduce Morten Allentoft, please. Morten's the leader of the Trace Environmental DNA Lab, a trend lab, in which this program's hosted at Curtin Uni. Morten's going to be giving you a brief introduction to the program and the outcomes before we get into the speakers. Over to you.
Thanks very much, Shane, and thank you everybody for showing up here today. It's so wonderful to see so many faces, and dare I say familiar faces. Now I've been in Perth for 3 years, and I'm actually starting to recognize some of you. For the first 3 years I've been faking it, just so you know, but now it feels genuine. So thank you very much for spending this lovely afternoon with us in this eDGES showcase. And Shane said, you said it's 5th time we're having this. Wow, that's incredible. Okay, today we'll be talking a lot about environmental DNA. I bet all of you sitting in this audience here probably know what environmental DNA is. So I'm just going to do this very, very brief, but it seems odd to have a whole day about this discipline without at least mentioning what we're actually looking at. So in Trend Lab, as you all know, we focus, it's one of our focal areas at least, to use environmental DNA as a tool to monitor the environment. And all living organisms shed DNA into the environment when they die and decompose, when they urinate, when they defecate, when they breathe, whatever. And we can apply sensitive DNA capture methods called extraction to actually capture these molecules, sequence them, and then because all living organisms have a unique genetic profile, we can match those genetic sequences to online databases, right? And then tell which organisms are present in any given environment. And this is what we specialize in, and that's of course a wonderful tool to monitor biodiversity around us. No reason to dwell more on that. You'll hear a lot about environmental DNA in these various talks after me. A few words here about our actual eDGES program, which is of course supported exclusively almost by BHP's social investment framework. We are housed at Curtin, so you can say Curtin is providing our facilities, but everything else that you hear about today is exclusively supported by Tim and BHP. So that's of course something we value tremendously. The overriding aims is to seek and understand ecological evolutionary change in natural and man-made landscapes. This is a very broad brush aim, you can say, of the whole program as such. When we have that understanding, we can develop and apply eDNA technologies to address biodiversity loss. We also aim to then take that knowledge and develop new generation tools for improved conservation. So it's not only about understanding and monitoring, we actually want some concrete outcomes as well that goes into direct conservation management. And then a lot of these studies are based on whole community studies. It's not just single species. We do have a few of those as well, but we're using often most meta-barcode approaches, which can profile the genetic diversity in the whole ecosystem, or at least in certain subcomponents of the ecosystem. And I've already said that this is, of course, supported by BHP. We had eDGES 1, version 1, it ended last year. It was a 4-year project. There were 7 sub-projects that were funded with 2.9 million from BHP's social investment framework. There was a team of around 10 people in Trend Lab. And now eDGES version 2 kicked off last year. It was presented at our last meeting in town, this same workshop. And it's, again, a 4.5 year. Hi, Ewan. 9 projects. As you can see, both the budget, the number of projects, and the number of people has actually expanded now. So we have quite a substantial group of people working together under this eDGES banner. Our motivation is, of course, to create new knowledge. That goes for any sciences, right? So that's very broad brush, but we also want to translate that into concrete outcomes. And all the projects you're seeing today gives you examples of how we're creating new knowledge, but also how that's then that is hopefully at least going to be applied directly. We have listed these 4 primary components up here that are at least some areas of focus where we believe we are delivering. We are delivering high impact research and peer-reviewed papers. That continues to be a motivation. We are a basic research lab, and this is a fundamental research lab, and this is our bread and butter, right? I mean, this is if you want to do translational science, you need something to translate. So that is our core research areas. We want to, of course, engage and communicate with the public, And you will see all these, at least several of these research projects that are being presented today have a strong educational element built into them. Facilitate global networks. Several of the projects here have already instigated some big networks and some big collaborations across the globe. I think in particularly Mattia's project on the Chilean wetlands and continuing in subterranean space have really facilitated some tremendous networks, and I think he'll be talking a little bit about that. And then I also want to mention that the whole eDGES program, it has really become an incubator for new research talent. It's becoming more and more obvious as this program continues, that it's incredibly valuable from that perspective. Perspective. I mean, as a head of a research lab, you often most see the talents leave. That's how academia is. Unfortunately, you train people, they become really good, they start to teach you something, and then they bugger off, right? I mean, and that's really. But eDGES actually allows us to keep some of the best talents that we have in our space at Curtin, I would say, and it's incredibly valuable for us to do that, because it ensures that we're not just losing the knowledge as soon as we have created it. And I would say this is becoming more and more clear to us that it's a tremendous privilege. Okay. Very briefly, I'm not going to go over this, but just to say that we continue to deliver high impact research, as I mentioned, just from the basic publishing paper perspective. We had 12 papers out just this year from our eDGES program, and I can't even remember how many we published throughout the whole program, but it's way more than 50, right? Possibly closer to 100 actually. But anyway, we are continuing to deliver in this space, and again, outreach impact. We have numerous, engagement every single year. This is, of course, kind of our flagstone event where we get everybody together and showcase our research, but there are many other examples, and some of these will be mentioned in the individual talks coming up after me. And then I just want to mention again that, of course, we are being supported incredibly generously by BHP and the social investment framework, but we couldn't do this without Curtin University as well. We have really great facilities, state-of-the-art molecular facilities at Curtin to actually conduct our research. And that goes both for modern DNA laboratories, but also clean lab, ultra sterile clean lab facilities where we can work with these highly problematic and highly degraded molecules that eDNA is, and particularly also that ancient DNA cells. And we're a great big group of people that work together in this space. And I also just want to mention this because eDGES has to some extent become so successful that it's becoming a brand in its own a little bit. So I get executive people now from Curtin telling me if they can come and see the eDGES lab or the eDGES group and meet them, right? And I just want to mention this. This is a research program that we are hosting in Trent Lab at Curtin University. So that's a little bit important. And along the same lines, I just want to mention that environmental DNA is, of course, I would say probably 80% of what we do. But we are actually engaged in other research avenues as well in this, whole discipline that we call molecular ecology. So we're using environmental DNA to see what is present in the biosphere to this day. We're using ancient DNA to go back in the past and trying to map out the present day, the past diversity to give that temporal context. And then we use genomics to try and understand the evolutionary processes that had shaped that biodiversity that we see to the present day. I have noticed that people at Curtin, they don't really know in general what DNA is, but they do know what eDNA is, and that's interesting. And I blame Mike Bunce for that. So that was my predecessor. But anyway, I think I'll just stop here actually, and then hand over to the various leaders of the different projects that will present this for you. So thank you very much again, and thanks for showing up today. Thanks, guys.
Right, Angus. I've already introduced you, but yes, so this will be the first ecological presentation for wetlands conservation. First up is Dr. Angus Lawrie.
All right, thank you, Shane. So firstly, I'd just like to acknowledge that this project is me and a lot of other people's whose name are on the slide. I'm just the one talking about this here today. So the Living Waters project is a very large component. It's a very large project. It has lots of different avenues. Because I've got 8 minutes, I'm only going to be talking about the first topic here. But I'd just like to reference that as well as the eDNA monitoring tool for wetlands I'm going to talk about. We're also interested in exploring how these wetland ecosystems have changed through deep time, and Dr. Matt Campbell is running a project looking at how ancient DNA communities in these wetlands. And we're also the engagement that Morten had up before has been part of the LiWA project. We're making a real concerted effort to try and talk to different stakeholders and community groups about environmental DNA in a wetlands context as well. But the main focus of certainly what I do is trying to develop a eDNA monitoring tool for wetlands. And I'd actually just like to start by giving more context. So wetlands come in a range of sizes. They can be large, deep, and permanent, seasonal, temporary, and shallow. They can be reed dominated. They can be plant dominated. They can be open water. They can be small. But what they all have in common is that they're all critical in landscape contexts. They support unique biodiversity. They support endemic crayfish, different frogs, different snake-necked tortoises, different frogs. We did a lot of frog work last year. But they're also some of the most at-risk ecosystems as freshwater ecosystems. So they're experiencing profound drying, salinisation, exposure to different agricultural pressures, and effects of urbanisation. So these ecosystems are both incredibly important in our landscapes, but they're also extremely at risk. Like anything else, we need to monitor this change. And I just make a general point here around how we develop biomonitoring tools in a more general context before I relate it to actually what we're doing. Essentially, what we do in biomonitoring is we make associations between types of environments that we go to and the animals, plants, whatever, that exist in them. So in this context, we might go to a lake and find that it's full of fish and frogs as an example, and deduce that it's in good condition, go to another lake that is of poorer quality and see that its composition is mostly worms or snails or what have you, more hardy taxa, and decide that that lake is of lesser quality. Essentially, all we're doing there is making an association between the animals or plants that occur there and where they occur. And why that's really useful is that we can come along and use environmental DNA, so we can replace these animals with actual DNA sequences without necessarily knowing what that animal actually is, and replace these traditional indicators of ecosystem health with DNA. And why that's useful, particularly in the context of environmental DNA, is one of the major problems we face in environmental DNA is there's a lot of biodiversity, and unfortunately there's not many taxonomists that have actually told us what that biodiversity is. So we only know what a small fraction of the DNA sequences are when we conducted the eDNA analysis. We only know what a small fraction, what those animals, plants actually are. So this is real data. This is just a representation from one of our lakes of the proportion of DNA sequences where we know what kingdom it belongs to. This is extremely high taxonomic level. And this is the total number of DNA sequences that we actually collected. And so what we're doing as part of this project is trying to understand how could we use that whole pie chart rather than just that small fraction. So essentially what we're trying to build here is a DNA index of the whole pie chart, all the DNA sequences. And so this project, initially in 2024, we did a preliminary study of 65 sites. We sampled from all across Perth. We sampled lakes of varying quality, from good lakes to poor lakes and everything in between. We didn't just collect eDNA, we contextualized our eDNA data with a range of other metrics, more traditional biomonitoring metrics. And then we blasted our eDNA samples with a range of assays, which basically tell us about the biodiversity from a micro scale all the way up to the big stuff, to the plants and the animals and the invertebrates. And I don't have time to tell you how it works, but trust me, it does. So preliminary results essentially suggested that wetlands that we'd expect to be bad, so wetlands that have high nutrients, high invasive species, and no riparian vegetation. So riparian vegetation is the vegetation around the edge of a lake. It's like the eyelashes that stop the dirt getting in. We found that those lakes, generally speaking, tended to score poorly on our index that we developed from our eDNA data, but we found that the lakes that we'd expect to be good scored better. So these results were really promising. But this was only for a small geographic region. This was just from around Perth. And so what I've been doing over the last kind of 4 months is going across the whole of southwestern WA, so massively expanding our geographic coverage to encompass everything from Kalbarri to Esperance and everything kind of in between from a freshwater ecosystem perspective. So we have a total of 172 sites. So this is now a very large data set. I don't have any results to show you, but it's just to point out that we've now expanded this project to include there's quite a few European countries that fit within that geographic scope. Like it's a very large data set, and hopefully we'll be able to say something about the ecological health of wetlands across that entire scope. But for the next 2 minutes, whatever I've got left, as well as understanding, can we make a model that actually deduces health across a broad ecosystem scale? We also wanted to know how does this index change through time? So we're doing an ongoing study where we're monitoring 11 sites across the Perth region, trying to understand how these wetlands change at a monthly scale. So this has been going on since April. I don't have all the data to show you, but I have a couple of examples. And this will go on until next April. So it will be 1 full year of 11 sites sampled monthly. And its wetlands are permanent, so lakes that have water all year round and lakes that also dry out every year. And our questions are, how does the actual index change through time? Does it matter if you collect a sample in August or if you collect a sample in October or in March? How does it actually change? And just as a bit of a taste of what that data kind of looks like, this is only for a few months because we're as you can see, we've done a lot in this year. This is just for a few samples, but this is one lake called Banksia Lake that's in Coburn. So in April of this year, we sampled it and had this much water in it. It was incredibly saline. And then this lake dried out about a month later. So we got it at the very end of its hydro cycle. And then we sampled it again and again and again. And then we sampled it, for example, in September, and it looked like this. There's about 2 meters of water there. So this ecosystem has fundamentally changed. How did the aquatic score, which is our index, change? What we saw is it generally starts to increase as it fills up with water. And I don't yet know what this means because I haven't been able to think about it too much. But my main takeaways from this is that the index doesn't fluctuate massively between the months, which is probably a good thing. It means it's relatively stable. But what the representative change between one score and another, I don't know what that means yet. But it's stable, so that is a good thing. And generally speaking, it's doing what I would hope it would do, in that when it's very shallow, you would say poor. It's lower than when it fills up and it's higher. Another example, this is Lake Jandabup in the northern parts of Perth, and this is to say that we got the same point. Generally speaking, as the lake fills up with water, we tend to find that the health score tends to increase. Although again, I don't know what it means yet between the differences between the health scores because it's an index. But that's all part of the fun. That's what's next. And then just finally, what we're doing next for this project in 2026 is basically trying to see whether or not we integrate all of these new data points that we have into one model. Can that tell us about the whole health of the wetlands in the whole of southwestern WA? We're continuing our temporal study, and we're also doing a study that's trying to understand, if we take a sample from one spot in the lake, how does that differ from other spots in lakes? So how does the spatial variability of this index actually change? And hopefully we'll output a really robust way to understand health in wetland contexts. And that's it. Thank you.
Fantastic. Thanks, Angus. We've got one question online, and then I'll go to the room.
Thank you very much. I've got a question from Ollie Berry, and he says, how do you account for biogeographic structure in taxa over a broad area?
Excellent question. So that was one of my main reasons why I limited our initial analysis, our preliminary analysis, to one biogeographic area. And that was because for that exact question. So if you can imagine that if I went from Perth to Esperance, we would change biodiversity communities just because we'd traveled a distance, not necessarily because we'd changed anthropogenic disturbance necessarily, which is basically what we're trying to measure. And there's two answers. The first answer is we don't seem to see an effect of biogeography based off the preliminary data that we have, mainly because the way that we choose our indicator sequences relies on finding the most commonly occurring sequences in a lake. And what we tend to find is that those really common occurring taxa are also commonly occurring across that whole biogeographic area. But secondly, and I think actually this is the clever bit about the people who came up with this idea, which wasn't me, DNA sequences can be conserved in evolutionary context. So you have an expression, we're 97% banana or something. We share bits of DNA with other things across evolutionary lineages, right? So for example, we might pick up a dragonfly in Perth, we might pick up a dragonfly in Esperance. They might be different species, but looking at the specific gene we're actually measuring, they might be exactly the same. So their ecological function might be exactly the same.
Excellent. Any questions in the room? Sam.
Thanks. Yeah, I'm just curious, like how many sequences you think are actually driving, like how many different species are actually responsible for driving that metric?
Yeah, so it's a good question. So at the moment, our model contains 5,000 sequences, indicator sequences, but the number of indicator sequences that you can choose based off the way we do this with an unsupervised learning algorithm, you can choose as many as you want. And there is a level of redundancy at some level, but there's one of the things that I'm thinking more about, and I'll stress that this isn't finished. But one of the things I'm thinking about is if you've got a large number of sequences to choose from, do you have inherently a greater chance of centralizing to one number in a site because of just central limit theorem, right? You have lots of samples, so you're going to, your calculation of the average is going to be more precise just because you have a greater number of sequences. I'm not sure if that makes any sense, but right now we're using 5,000. We could use 1,000, we could use 10,000. I'm using 5,000 for various specific model optimization reasons.
Excellent. All right. Well, we might move on. Thank you, Angus.
Next up, we have Dr. Nicole White, who'll be talking on eDNA for subterranean fauna detection and conservation.
Hi, everyone. All right, I was told I'm not allowed to move around because of the microphone here, not microscope. So I'm going to do my very best. I'm actually going to hold on to the lectern here to keep me in place. So today I wanted to share with you, and my biggest challenge really was to capture the breadth of the amount of work that we've done in the subterranean space in 8 minutes. All right, so I work with the team, so I'm presenting not just on what I've been working on, but also what the subterranean team has been doing in the eDNA space. So these lovely animals up here are nicely illuminated, but if you remove those animals, that's basically what we're doing. We're working in the dark. We're working underground. We're subterranean. There's not a lot of light down there. And basically, we have so many questions that we want to answer and want to know if eDNA tools can help assist us characterize these systems. So what I'm going to do, I'm going to briefly talk about what I have been specifically focusing on, but how my project has also been working in collaboration and feeding and assisting other research programs in the subterranean space. Our ultimate aim is how can we enhance this molecular toolkit for subterranean fauna. So there's some PhD work, my collaborations with Michelle Guzik, who's in Adelaide, who couldn't attend today, but I'm sure she's online. Jake, who's also in Adelaide, I'll be speaking a bit on his, and Julia's project as well. So we're trying not to restrict ourselves so that we can actually capture the breadth of subterranean diversity. So I've done some work in South Australia on the Gab Springs. That's been very interesting. And this last year, I've been focusing on Ethel Gorge. Some of the other projects involved under this subterranean umbrella have extended across the Pilbara, and I'll be going into that. So really what I've been doing is shotgun sequencing subterranean animals. For those of you who don't know what shotgun sequencing is, it's slightly different to this PCR approach and primers. So when we're referring to a barcode, that's this standard little region here that we call a reference barcode. And when we get into eDNA, that barcode even becomes shorter because of the degraded nature of the environmental DNA. What I've been doing, because I've been getting a little bit frustrated with the limits of some of the existing assays for capturing the diversity of subterranean fauna, and we're talking about reference databases, I've kind of been throwing out the primers and going, well, what if we just take an animal, extract its DNA, and sequence the whole lot, the whole genome? So that's what shotgun sequencing is. So I've been working on very small animals to produce basically 96% more data than we can get with a barcode region. So I'm doing the complete mitochondrial genome. So this is how this eDNA and genomics is kind of now bridging in this project. What I can say is the success rate has been at about 50% from obtaining an animal and then actually being able to produce a mitochondrial genome. There's some technical challenges in that, but the biggest one, if you actually see the scale bar on the bottom of that image there, you can see those animals are only about 2 millimeters. So that's not a lot of starting material. When I refer to starting material, that's DNA to work with to build these shotgun libraries and generate these. But I'm actually really happy because I wasn't sure how well this was going to work. So now my lesson learned from the previous years is when I'm going to be working on these animals, I always ask for multiples of, because I'm expecting about a 50% failure rate so that we can capture them. So that's kind of what I've been busy with personally in the lab. Now, one of the outcomes of generating mitochondrial genomes, because it's not just these bugs underground, we actually have 3 vertebrate species that live strictly underground too, here in Australia. One of them is what we call this cave gudgeon. And from the mitochondrial genomes, which the museum kindly assisted me with getting tissue, I was able to shotgun sequence. And then we were able to design assays that are now specific for the gudgeon. So Milad, who's a PhD student in Adelaide, which I'm supervising, he developed an assay that targets the 16S gene. That's a gene on the mitochondria. And at the same time, or a little bit later, I've developed an assay that targets the control region. That's another gene on the mitochondria. So notice I haven't even alluded to the CO1 barcode gene, because when we had the opportunity to look at the total specific assay on. So this is the advantages and the kind of the outputs now that we're able to do. We're able to design assays and validate them. So what we can see, there's slightly different sensitivities between the 2 assays, but they are now well validated for both the Cape Range and the Barrow Island gudgeon. And I should have my paper submitted by December. So that work's now written up. So I'm going to move on now to Michelle's work and what we've been doing. So Michelle's been working really hard to kind of wrangle in what do we do when we do all of these barcode sequences, and how can we get a central database in place for working with these barcodes. Because when we collect environmental samples, we are heavily reliant on a database that we can compare the DNA sequences to. Therefore, we can say we found species A, species B, species C in this underground system. So curation of these databases is really important. And Michelle has proposed in a paper that basically the proofs just came out on Friday. So that's towards a global barcode reference library for subterranean fauna. And in that, Michelle has proposed a 4-stage plan, as we can see over there on the screen, for developing the data infrastructure for subterranean fauna. So if you're interested in that, that paper will be accessible very, very shortly. In the meantime, to doing all that, we've also written and submitted another ARC linkage grant. We have more questions. We do research, and then we tend to find we have more questions come up that we would like to answer. And we got very, very good reviews. And we are just waiting the outcomes of the decision on that linkage grant, which should hopefully be early in the new year. And then we get to expand our subterranean portfolio even more, because we have 3 PhDs and 2 postdocs written into that linkage grant. So fingers crossed on that one. So I'm going to move over to Mika's work. So Mika is now on maternity leave. So she's done another project. But before she left, she was working really, really hard to validate existing assays. Like, how well do the existing assays work in subterranean fauna? So she could take that custom reference database. She looked at 31 primer combinations and then started testing the sensitivity and then also the specificity. So what was really interesting is some primers looked really promising, but in environmental DNA and subterranean systems, we know there's not a lot of DNA, and some of these primer sets, even though they looked really good in silico, they bind, they actually required a lot of copies of DNA to get that PCR reaction happening, which isn't often what we find in subterranean systems. So where that red arrow was basically the champion of what's already published in the literature. Now, we also know from this work, certain groups that all of these assays, they don't pick up. And that's more work for us now to do. So we've got the mitochondrial genomes. We know what groups don't currently get picked up, and that's the next steps with the work. Now I'm turning over to Jake's project. Jake's now a PhD candidate at the University of Adelaide that I'm also supervising. And similar to Jake, he's using a taxon-independent analysis for trying to develop a biotic index for groundwater systems using microbes. And we're actually seeing some separation here from low impact, moderate impact to high impact. And very lastly, Julia, who's now left Australia, she's an expert taxonomist. She's been busy describing species, and then also I've been doing the shotgun sequencing and generating mitochondrial genomes for those. So thank you very much, everybody, for having a listen. I've been told now I'm cut, but I really believe, we are a very, very small subterranean team, but I believe we're punching well above our weight, and we hope to continue to do a lot more research in this area. I didn't talk about Mattia's work because he's coming straight in now, so I didn't want to steal his thunder. Thank you very much for listening.
We can fit in one quick question if anyone listening.
And so
You're released, you did well. Yes, paid for you.
All right, excellent. Oops, here we go. Okay, so next up we've got Mattia, and he's...
Hello, my name is Mattia Sacco, and I'm the researcher in charge of the so-called eDGES Project 3, together with a set of collaborators at Trend Lab and also international collaborators. Today I'll be talking about hypersaline ecosystems and groundwater environments and how eDNA approaches can unveil crucial biodiversity patterns. I'll start with a recap on the eDGES Project 3 version 1, then I'll move into Project 3 version 2. We'll be focusing specifically for this one on Serra de Punta Negra, and I will outline sampling plan and goals. And I'll finish with an overview of the so-called GREG initiative, standing for Global Research on eDNA in Groundwaters. We have 3 main goals there and mostly two phases, and I will outline structure and network of collaborators. Let's have a look now at the eDGES Project 3 version 1. With this project, we were able to fully fund a PhD project at the University of Antofagasta in Chile. The student, Gonzalo Salazar, is actually due for thesis submission in March 2026. So hectic time for him, as we can all imagine. Paper under review in freshwater biology, plus 2 papers actually in preparation, and he was also being able to lead a review recently published in PJ. The picture on the left summarizes all the systems that were part of this review, 63 hypersaline lakes in 4 countries. The review mostly focused on macroinvertebrate diversity, 46 taxa in total. To our surprise, somehow, location or ecosystem size has no effect at all on the patterns observed in terms of ecological dynamics. and contrarily, variation in community structure were mostly driven by salinity. Another work in progress is a paper on the use of eDNA for microorganismal diversity. For this one, we focused on 3 salars, Salar de Puxa, Salar de Tara, and Laguna Puilar. Once we had a look at the water diversity, from a microbial perspective, we saw that there was a clear dominance for these 2 groups, which are actually common in hypersaline lakes. And once again, salinity was a major driver in shaping deterministic processes across these 3 lakes. As I said before, eDGES Project 3 version 2 will focus on Salar de Punta Negra, which is relatively extensive. 14 to 30 millimeters of rainfall per year, so not much rainfall, surrounded by deposits left by dry valley, so the water is quite occasional there, but there are some springs where the water is actually permanent. So Salar de Punta Negra is very interesting from an ecological point of view because we have the so-called VC-AL system. We have a laguna which is hypersaline, connected through a canal to a spring which is mostly fresh water. So one of the research questions that we want to target is that is biodiversity matching this hydrogeological pattern in terms of richer or poorer communities in these 3 parts of the VCL system. Industrial operations on site have unfortunately disrupted the hydrogeological balance of the salar, and there's ecological restoration plans underway, and this project is one of them. Sampling plan at Salar de Punta Negra will involve, of course, eDNA analysis from water samples in the VCL systems. We will focus on the entirety of life, from bacteria all the way to wading birds. Choose sampling campaigns in 2 contrasting seasons, and there's a strong partnership with BHP collaborators on site, Magdalena Fernandez, Matias Perez, and Silmar Acosta. Potentially, we will include an additional site for comparative purposes, but mostly I wanted to talk about the 3 main goals in this slide. We want to investigate the community patterns across a marked salinity gradient and under contrasting seasons. We want to expand biodiversity surveys beyond routinary approaches. And last but not least, we want to provide molecular data for a long-term plan, which is potentially extendable to other systems such as the Salar de Pajonales. As I mentioned before, the second part of this speech will focus on the GREC project. I dropped the link over there if anyone is interested. We have 3 main goals. We want to ambitiously provide the first global eDNA study for subterranean biodiversity, and again we will have a tree-of-life analytical approach. We want to investigate the role of environmental drivers at an unprecedented large scale, and how these drivers shape subterranean biodiversity. And we also want to harness global eDNA outputs to raise awareness on a hugely overlooked ecosystem, which is groundwater. We are currently in the middle of phase 1, which is self-funded. For that reason, we focused on biodiversity hotspots and a closer set of collaborators. And we will produce preliminary data and tests. These lessons learned will be very useful for applying for a grant, large-scale grant, which will involve citizen science and a deeper focus on the global south. I dropped there the screenshot of a paper that outlines the characteristics of these 2 phases if anyone is interested. As I said, vast set of collaborators. We were able to gather a vast group of researchers and friends all over the world. And the way it worked is that basically we shipped a kit to all these collaborators. This kit that we designed contained 2 filters, buffers, gloves, and also protocol sheets. 50 plus collaborators actually from 5 different continents, and we targeted 45 different biodiversity hotspots, including karsts, alluviums, and alkaline caves. This, as already stressed, is a highly collaborative project, which involves the expertise, and we're very lucky to count on that, of this Dr. Michel Gudzik, Dr. Catherine Corbell, the GREC project manager Abdullah Masri, and the PhD student Mahir Matawal. So overall, there's 3 universities involved: University of Adelaide, Macquarie University, and Curtin University. I just wanted to conclude by giving an overview on the timeline of the project. We gave on the 5th of June 2025 an online seminar to present the initiative. Then we involved more and more collaborators until we closed the cohort of collaborators for this initiative, especially for the phase 1, given it's self-funded. We shipped all the kits and we are expecting to receive them back by the 15th of December this year. So hopefully by mid next year we will have all the results and the manuscripts will be in preparation, especially because we have planned for the 17th of June an in-person seminar at the Natural History Museum here in Paris, where I'm actually based at as a visiting researcher. An event, this workshop, that will also be funded by the Curtin Global Collaboration Seed Grant. And with this I conclude, and I thank you for your attention.
Hey, thank you very much, Mattia. That's a really impressive collaboration there just at the end. Okay, so yes, Mattia's not here, obviously, so if you've got any questions, then please get in contact and we'll pass that through to him. Next up, we're changing themes to the marine conservation. So I'd like to welcome Professor Simon Jarman, who'll be talking on 2 projects. So the researchers that lead these 2 are actually away in the field at the moment. So the first one is detecting invasive marine species, and the second one is mapping life in the northern Pilbara.
Great, thanks, Shane. So the first project is about improving sensitivity of eDNA methods for detecting invasive marine species. So a lot of you will know what invasive marine species are or have some examples in mind, but essentially they're any organism that can move from its native range into a new range and establish itself there. And they come from all animal phyla essentially, and most plant phyla, and there are some unicellular examples as well. So some diatoms and dinoflagellates can be invasive also. From everything you've heard about eDNA, it's obviously a very good method for approaching this very broad-scale problem of being able to identify things from such a wide range of things. If you're using microscopy, you'd miss some of the large things. If you were using visual underwater cameras or something like that, then you would miss some of the microscopic things, whereas eDNA can get everything in one test. That's the basic idea and why we apply eDNA methods to this. Age of these organisms. So the critical life cycle stages for many invasive marine species are the larval and egg stages. So if you can detect things when they're establishing in a new environment, it's more important almost than detecting the adult stages, or equally important. And eDNA does that very well. So you can detect all these things that quite often even aren't recognizable morphologically as being the adult stage at the larval stage, like all these larval crustacea mollusks shown here. Or that's a starfish, as you can probably see there. You can detect them at that stage, and this is great for IMS screening. eDNA is also a very sensitive method. It can detect very small amounts of material, but this does lead to a problem in that you get lots of false positives in these testing regimes, because you can have small amounts of material that isn't viable that you do detect, or you can get small amounts of contamination that's detected in these assays. And part of what I'm going to talk about today is dealing with these kind of problems. But just before I go on to that, we've recently completed analysing some work that we did a couple of years ago, surveying the Port Hedland area, where we took about 1,000 samples to look at eDNA in that region from these sites that are numbered on this map. We screened all of this for invasive marine species. We found one thing out of all of the things that we looked at that may be an invasive species. It's not a perfect match, but it's a close match to this invasive barnacle species that has been known as an invasive for a long time. It was first detected in the Netherlands in the 18th century, but is originally from the northern Indian Ocean. But because we're detecting this stuff in the southern Indian Ocean, it's entirely possible, because it isn't a perfect match, that it's an endemic that hasn't been identified yet. So if you know any barnacle taxonomists, or any of you are, get in touch, because these are the sort of things that these products show up. But anyway, that sample, that species, or that hit, did show up in 17 samples out of the survey. So it's clearly something that's there and something that we should follow up on anyway. But beyond that, there were no other invasives that we could detect in the Port Hedland area, which is actually great news. It was a very thorough survey. So just moving on to some of the other reasons that we might want to improve methods for studying invasive marine species with eDNA. The main way that we do it currently for these really broad-scale screens is to use DNA meta-barcoding. So this is the very broad-scale kind of approach that has been spoken about a lot already. And this is a great method, but it has lots of imperfections. So one of the main problems with it is that the primer sets that we use, the PCR primer sets that we use, try and get everything, and in that process miss some things, and we quite often can't work out what they are missing. So this is actually this primer set. This is from the Lawrie CO1 primer set that's used quite often. A lot of you who've done eDNA work will have used this primer set. This is from their paper. This is an analysis of how conserved one of the primer binding sites is, and this is one of the primers with just coloured bars giving you an indication of how conserved it is across that region. As you can see, it's a complete mess. Like this primer is 256-fold redundant. So when you make this, when you synthesize it, you actually get 256 sequences back for it. Almost none of them are a perfect match to the targets that they're going for. And they acknowledge this in the paper. This is their analysis of how often it misses things in the paper, with 0 misses over on that side, and for a different phyla. And almost all of the cases, it has multiple misses of things there. And this isn't necessarily terrible. It just means that you have a biased representation of things. Some things will be overrepresented in the data set as a result of this technical problem. But we can get around this in some situations, and I'll tell you why in a minute. One of the other reasons that metabarcoding can fail and can be problematic is that every detection you make in an eDNA sample with metabarcoding is dependent on everything else that's in that sample. So just to illustrate this, if you were looking at fish in this lovely coral reef area and you were just interested in the fish, if you analyze this community, you would get something like this with the sizes of the squares corresponding to the abundance of the fish. If one fish has a spawning event where it throws huge amounts of gametes into the ocean, which are represented by red here, what happens is that everything else gets squashed up. That dominates the sample. And at some level, there will be a detection minimum that you have to pass to have your eDNA sequence represented in the data set. So things that don't actually change the underlying biodiversity, so the fish community is actually unaltered apart from that spawning event, can affect the detectability of other things in the data set. So this leads to false negatives. So false negatives are a problem. Another problem is that for metabarcoding experiments, the only real way that you can have a positive control that's really acceptable is to put some control DNA from things that you're hoping to detect into the sample and maybe mark it with some changed molecules so you can say it's control DNA, and then detect it later on. And that's okay if you've got a few species that you're interested in, but you can't do it for everything in the ocean. It's just not possible. So there are too many things there. So we've got this great method, but it has some restrictions, and these restrictions lead to interpretation problems. Another major problem is the fact that endemic species may be close relatives of the IMS species, like the example I just gave at Port Hedland. And these are my colleagues, Shirley Lucas and Fred Wells, who are also involved in this work, who are currently in Southeast Asia, currently in Singapore, collecting samples from relatives of Australian invasive marine species so that we can solve this problem for some limited ranges of groups. So we need to have that reference material from nearby species that may be invasive in Southeast Asia and species here, so we know what's going on. But why do these false negative and positive problems matter? Well, basically because very expensive decisions get made on the basis of eDNA detection results. So you may have issues with decommissioning large marine infrastructure, or whether ships can move from one area to another. And IMS testing should ideally be a yes/no answer, right? Or at least have probabilities associated with it so that we can make more informed decisions. So given that, and given the sources of uncertainty that we have from this very broad-scale IMS testing, we've been working on some alternative methods for IMS detection that will hopefully get around this, but get around some of the problems of the expensiveness of single species testing. So there's a whole separate set of tests, single species testing like quantitative PCR, which a few people have mentioned already in their talks today. That's the kind of canonical method that can actually give you recordable rates of false negatives and positives. You can basically fill out a confusion matrix, a contingency table like this that will give you all of those methods, and then you can do more extensive analyses like ROC curve analysis to actually identify the characteristics of the test. And what this means is that you can get probabilistic outcomes rather than a vague outcome of, yes, it showed up in the list or it didn't and we don't know why, which is sort of unsatisfying. But the problem with those single species tests is that we can't apply it to everything because it's too expensive. However, if we look at some situations like the invasive marine species test list that the federal government comes up with here, so it's different for every state, but this is the Commonwealth Government's list for priority marine pests, and it's only 20 species long. So that actually becomes quite tractable. With only 20 species, you can't quite do qPCR tests for all of it. It'd still be too expensive. But there are some approaches we can take that will actually get all of these things and solve some of the other problems. So I've been working with Cheryl primarily on developing a method that is not like the standard metabarcoding where we use one primer to try and get everything and it doesn't match all of the sequences properly. But we divide it up into a bunch of genus-specific primers that will get all of the species on that list and their close relatives, so we can deal with some of that problem with local endemics. So we basically divide it out into all of the genera that are present, and some of them can double up. So if we go back to that list, I think there are a couple of mollusk species, maybe the Perna species that has two components within it. So anyway, we can reduce the number of tests. So we've been working on this. Basically, we've been going through that list of priority marine species for Australia. We've designed primer assays for all of them. We've completed that phase. Cheryl tested 16 of these primer pairs in singleplex. They all work like that. And we tested them in one big multiplex PCR for 10 of them. And this works. So, and this is quite interesting because there's a sort of a dogma in dealing with metabarcoding that you can't multiplex metabarcoding primer sets. And if it's a full metabarcoding primer set where the taxonomic range is really big, you can't. It's true, because the PCRs are too complex, everything interferes with each other. But if we break it down into these much more specific primer sets, it really does work. So Cheryl has demonstrated that works. We're just working on the second multiplex now, and the idea is that in time we will, sometime during next year, we will have a test that will identify any of these marine pests, and you'll be able to assign probabilities to the outcome. So we can test all of these things with a proper contingency table kind of approach. The other things we're doing in this project is we're also developing an in-field testing procedure, which will end up being something very like the lateral flow test, which everyone here will remember, of course, from recent history. And basically, you'll do a bit of a pre-processing step that gets an appropriate DNA signal, and you'll put it on the lateral flow test, and you'll get a yes, no, and you'll be able to do that in the field. The multiplex IMS test should be ready later in the year, and I'm really excited about this because if this approach works, and we can do it for lots of other situations, it gives you a management-focused way of designing your assays. So you can come up with a management problem like, can we identify these particular IMS, and what probabilities can you assign to it? Following what we've done, the answer will be yes, you can do it, you can come up with these tests, and it will give you something that in the long term you should be able to make better environmental decisions on, or management decisions. And the other thing that's going on in this project is some sampling of invasive marine species, both within Australia and their near relatives and in Southeast Asia, because it's the area that most of our IMS are most likely to come from. So that's underway. So that's my conclusions for that project. And I might, if anyone has any questions on that, I might take those now. And then I've got another project to talk about that's kind of related.
Simon, less of a question or more of a comment. I'm from the WA Museum. I head up the science team. We absolutely have barnacle experts at the WA Museum.
Great.
We have Di Jones, who described over 90 barnacle species, and our current curator of crustaceans, Andrew Hosey. So yeah, if you're looking for taxonomic expertise, please reach out. I'm more than happy to help out.
That'd be fantastic, Jen. I think we should follow up on that one because it's an interesting situation. It's got some good history associated with it.
have another online question also from Oliver. Will you be vouchering these overseas invasive species in museums?
I don't know. So the team who are doing that are all overseas at the moment. I assume that they will be. Yes, I don't know, but I could get back to you on that.
Very much.
Questions?
Okay.
Part two.
Well, I'll just move on to the second part. This will be a very quick talk. So I'm basically talking for Fred Wells and Shirley Lucas because they're in Southeast Asia collecting invasive marine species. So they couldn't be here today, but they have this project on biodiversity mapping of the northern Pilbara coastal marine estate using environmental DNA. And it's not just environmental DNA. They are collecting lots of eDNA, but they're collecting lots of morphological samples as well. And this is a great opportunity to voucher some of these specimens of IMS and potential IMS. But the main thing that they've been focusing on is this group called the Oncodiidae, I'm going to say, which are sea slugs that aren't nudibranchs, essentially, is the way I'm going to describe it. So these are air-breathing mollusks, and they're one of these great reinvasions of the ocean, sort of less famous than whales and things like that, but it's the similar kind of idea. So they're not nudibranchs, but they're air-breathing slugs that have gone back to the sea. They're particularly abundant in the Pilbara. So it really makes sense to study these there because it's a biodiversity hotspot for them. Hardly any had been described before Fred started his work there. And basically they're doing a lot of species discovery in this area. So they've been sampling all up and down the coast from Carnarvon to Broome. This is a picture of what some of these things look like in their natural environment. So little brown blobs on a mud flat like that, or you find them in the mangrove roots and so on. And this is great because this is the sort of biodiversity that a lot of us are unaware of. I've never heard of them before. Fred brought them to my attention, so it's great to hear about them. He's been bringing international experts in to work with him on this. So Benoit Diat has come in from the US to help him with this because he's another taxonomic expert on that group. They're basically going to these regions, photographing them in situ, collecting lots of samples, and taking both eDNA and DNA samples from this group. So the results so far are that they've got 9 species in 3 genera catalogued for the region. So this is full descriptions. Some are widespread, so they're sort of from the whole Indo-Pacific region, and others are known only from Australia so far. And some are new species, which are in the process of being described. And Fred's just been working with an Aboriginal corporation there to try and name one of these new species and give it an Aboriginal-based name, which is fantastic, of course. The other big thing that will come out of this project is Fred is writing a book on the marine environment of northwestern Australia. So chapter 1 will introduce current Northwest Australian biogeography, go over coral reef biodiversity, various other reef diversity things. So Fred's very interested in mollusk reefs as well, and all the other specific things about that area that are interesting. So the big tides, the tsunamis and cyclones and so on that have affected it in recent years. And the following chapters will go on to talk about specific faunal and floral aspects of that area. And it's scheduled for completion at the end of 2026. So just to summarize this project, mollusks are one of the most diverse marine animal fauna, of course, and a lot of IMS come from mollusca. It's a big group for it, so it's really worth doing it. Northwest Australia has a particularly rich mollusk fauna, and a lot of it is undescribed. So it's a really good opportunity to actually describe lots of these species in this area that is essentially funding this project through BHP, which is wonderful. And it's going to be summarized in a book which will be accessible to everyone and will describe this for not just scientists but the general public as well. So that's all I have on that project. Thank you.
Thank you, Simon. Does anyone have a question for Fred on behalf of Simon? Yeah, I'm wondering. Okay, we might
go good.
Okay, great. So we're going to change themes now. So we're moving on to the terrestrial biomonitoring theme. So we've got 2 speakers in this group. The first one will be by Dr. Marina De Oliveira. She'll be talking on broad-scale monitoring of land systems with eDNA. And then following her will be Dr. Josh Newton talking about improving reptile monitoring in the Pilbara. Over to you, Marina.
So hello everyone, my name is Marina. This is my first time presenting at this symposium, so I'm very happy to be here. I will be presenting today on behalf of the big team on the screen, and I will update you on our project called Broad Scale Monitoring of Land Ecosystems with eDNA. This project started last year, so right at the end of eDGES version 1, so a lot of the things that I will present here are still ongoing. Just a little background. So as you know, Western Australia is the largest Australian state. It represents a third of the country. It has nearly 3,000 kilometers from north to south, which is approximately from Perth to Herbert, just so we have an idea of scale. This huge land is a challenge for monitoring. At the same time, we have a huge mining industry in Australia with a lot of economic importance, and most of that is based in Western Australia. So given the footprint of mining, the high biodiversity and endemism in Western Australia, we need to find better tools to are for aquatic ecosystems. And methods are more established and developed, but not in terrestrial eDNA monitoring, especially for arid regions. That's why we need better tools and dedicated research in this land. So terrestrial ecosystems comes with unique challenges such as what, where, and how to sample, how to deal with the lack of water, which is usually a good source of eDNA, and working on large remote areas where species naturally occur at low densities. That's where we come in. This leads us to the three aims of our projects, which are optimize sample collection for remote terrestrial areas, explore the spatial and temporal resolution of biodiversity, and start working towards terrestrial ecosystem health index based on eDNA. So one of the newest approaches with terrestrial biomonitoring based on eDNA is airborne eDNA sampling. Airborne DNA consists of DNA from any biological sample that we can capture from air. One way to collect it is with mobile devices. So filters can be attached to vehicles in different ways, on top, on the side, it doesn't really matter. And it provides a faster and more dynamic way of sampling. In this example here, we compare passive air filters that were deployed for long and short periods of time with the mobile sampling. All the methods, they detect a difference in the vertebrate community between two habitat types, the agriculture and the woodland. But we didn't see any significant difference between richness and community composition. Another study, this one was not funded by BHP, was part of Josh's PhD project. So he drove a long transect from Perth all the way almost to South Australia, collecting air samples. Here we can see only the detections of the invasive red fox, which is important because it's an important predator of native fauna. So the green dots, they are GBIF occurrences. GBIF is an online database that collects occurrence points from many different sources. And the orange cycles, they show the detection intensity from eDNA. So we can see that both patterns, they align well, which demonstrate the potential of airborne eDNA for large-scale biodiversity monitoring. Another component of our project is the spatial study. So we had two questions. If there are differences between sample types, and in this case, we had 3 different sample types, which is soil, that's the most used one, and 2 new methods, the passive filters and the rollers. And how far apart can eDNA tell the difference between 2 adjacent land uses? So this is our very happy researcher with her 360 samples that she collected from 2 different locations, one in South Australia, one in Western Australia, one is arid and the other one is semi-arid. 2 different habitats in each location, 3 different methods, and 3 different assays. The way we spread this sample, we place sampling points at increasing distance up to 500 meters from the neighboring habitat. We're still analyzing these results, so I won't show you right now. But we saw the eDNA can successfully differentiate between habitats regardless of the distance, but no single method was capable of detecting all the species, all the zootiles, all the sequence. So we need more than one method to capture this whole diversity. The other component of this project is a temporal study. So we want to see how species composition change over time and what's the best time to conduct surveys. We are doing this work across 5 different ecosystems. One is the Pilbara, and we're doing that with the help of the incredible team of BHP rehabilitation team. Thank you, guys. This project started in July this year and it will run for 1 year until 2026. All of that will help us work towards our final goal, which is developing an ecosystem condition index based on eDNA. So there are many ways we can assess the condition of the environment. We can, for example, look at satellite images and analyze the different lengths of the waves. We can visit places, we can collect plants, animals, anything, and analyze that. Or we can infer the condition based on species or sequences identified by the eDNA. And given the characteristics of arid and remote regions like the Pilbara, where we'll conduct our study, remote sensing in this case will provide us with the baseline for what condition looks like in this region. So we can start to shape this project. We had a workshop at Curtin in August this year. We had researchers from different institutions, from Curtin, from BHP, CSI, University of Melbourne, and Carey Institute. We started planning for the large-scale study that we are calling that way, and a review paper will come out of the workshop, gathering all the information available and all the knowledge that we gather in these 2 days. Overall, this project has so far 3 papers under review. We are conducting 4 projects at the same time. We have 460 samples collected, I think more now. We've presented the results at the Biodiversity Conference, and we are in collaboration with new research institutes and participating in community outreach events. For 2026, we're planning 2 fieldwork expeditions to the Pilbara. We'll have 4 additional temporal sampling across the 5 ecosystems. We plan to collect 1,000 samples, wish us luck. We have the submission of 2 other papers, and we'll strengthen our collaboration and engagement with the community. All of that so we can reach the impact and the future perspectives that we have for this project. And that's all for me.
Thank you very much, Marina. Difficult subject to get across. It's a great start. Has anyone got any questions for Marina?
Crickets.
Quick.
Slide that you're crickets. Are we monitoring them?
All right. Okay, thanks, Marina. Okay, so the next speaker will be Dr. Josh Newton to talk about how he's monitoring reptiles.
Soon to be doctor, not a doctor yet.
Close.
Yeah, everyone, again, I'm talking on behalf of obviously a lot of people that's part of the reptile project here. So I've been at Trent for a good few years now with honours and PhD, but only just started with eDGES, which is really exciting, all to do with the reptiles up in the Pilbara, mainly. But reptiles has been a part of eDGES since the very beginning, mainly driven by Ben, who's just down the front here, and his Pilbara olive python project. It's been a really great project, trying to detect the Pilbara olive python using environmental DNA. He's got a really highly sensitive assay. And he's also been testing lots of different sample types in the environment to see what is best at detecting the python, and also looking at the degradation of its DNA as well, and now looking at doing some shotgun sequencing to see if you can get more than just that kind of short DNA fragment
to get some more information on the samples. But we thought, Ben's been doing some chatting around town at different conferences, so we thought we'd give him a bit of a break. And I'd get up and talk and tell you some exciting things that we're kind of just cranking up right now. So you got me that's just started, and we also got PhD students that are literally starting this week, which is really exciting. But first, I'd kind of try to explain why reptiles in the context of environmental DNA, and trying to kind of prove to you that it's not just because Morten clearly has a bit of a love for reptiles, but there's a greater reasoning, I swear. Reptiles in Australia are really important. They're about 40% of the terrestrial vertebrate species, a lot of endemic species, a lot of endangered species, venomous species, elusive, cryptic species. So a lot of these things that when we're talking about environmental DNA that it's supposed to work really well with, it's supposed to be good at detecting these really elusive species. But in reality with reptiles, it's not actually the case. We have a lot of trouble detecting reptiles. So this is a pretty common kind of environmental DNA data set, trying to detect vertebrates, not bagging anybody else. This is my data set from a couple of years ago. You see in the blue, you've got lots and lots of birds. In the green, you've got lots of mammals. And for reptiles, we get a really small group, certainly not anywhere near 40% or close to that. And this is even more stark when you know that I actually sampled from within the reptile room at the Perth Zoo. There's an open air enclosure with 33 reptile species in there, so we're clearly doing something very wrong and not getting something right. And whenever we get data sets like this, what we write in our papers is that there's a hypothesis that reptiles don't shed much DNA. And it's likely true in comparison to your big dirty mammals and your birds with lots of hair or feathers and stuff like that. But isn't that what we do? Don't we work with trace amounts of DNA in the environment? So to say that eDNA doesn't work for reptiles just because there's not much DNA there, I think is a bit of a cop-out. I think it's actually us. I think it's our inability to sample the right areas in the environment to collect the DNA when we're out there. And then also we're not using the right molecular toolkit once we get to the lab. And that's what this project is really about, is trying to improve, one, how we collect the DNA in the environment, and then how we capture that once we get into the lab. So the backbone of a lot of this and where it's going to kind of start is we're going to generate 150 mitochondrial genomes for important reptile groups in Western Australia. This is in conjunction with Western Australian Museum, who's providing a lot of the samples, and CSIRO. And obviously because it's BHP funded, or at least a portion of this is BHP funded, we'll be focusing on a lot of Pilbara species, which are really important. This is hopefully just the beginning. There are about 600 reptile species in Western Australia. So ideally we can generate funding and we can do all of those species at some point, if anybody's interested in funding. But the big main reason for this is, I think one of the main issues that we have is there actually are no good assays for detecting reptiles yet, or terrestrial reptiles. So that'll be one of the first things that our PhD student starts on. It'll be starting to develop a reptile assay or multiple assays, so we can try, and once we get the DNA in the lab, we can really start to amplify that DNA and make the best of the samples that we have. The other side of it, and the thing that I've kind of been cranking up on lately, is we're trying to improve how we sample for reptile DNA. So I've been building, well, I suppose what we're going to do is we're going to do it in a control setting first. We don't want to just go straight to the field. So I've built 6 big 2 metre by 2 metre reptile Airbnbs at Curtin University. And we've just put our first reptiles in these. And the idea is we have those reptiles in there for 3 to 4 weeks. We let them do their thing, leave the DNA around the place, and then we take them out. And then we can start sampling all these different bits in the environment. So classic surveys for reptiles are cover objects. So they hide under things, so under different cover objects. We've got some filter paper, some sandpaper, some foam, things that might hold DNA better than other substrates, leaf litter, water, and places for them to hide and stuff. So the idea with this is we kind of gain a bit more knowledge on what's happening with that DNA in the environment, how long is it lasting for, is anything holding onto it, or can we extract DNA better off some substrates over others? Once we've gained as much knowledge as we can from this, we'll take it to the Pilbara, and then we'll kind of design ways to try and trap reptile DNA in the environment. Obviously, what we want to see ultimately is, can the insights we learn here, can we then translate that in field conditions? Because it's not necessarily going to be the same, but can we improve the DNA that we get in our samples? Because if we have no DNA in our sample, when we get back to the lab, there's nothing we can do in the lab to increase that. So you might be wondering, what are our first reptiles we've been put in there? Well, Morten and I have been having quite a bit of fun because we've decided that, or Morten decided that they were going to be Western tiger snakes. So we've been doing venomous snake handling courses over the last couple of weeks, which has been good fun. And then literally last week, we were out in the wetlands of Perth and down near Harvey capturing tiger snakes. Now, this is very natural to Morten, but this is not natural to me whatsoever. This is my first ever wild caught snake. And then we bring them back to Curtin Uni and we, excuse the audio, I don't know who's going to talk over it. And we're putting out the defibrillate snakes.
Oh, it sounds gorgeous.
He's just going to hang out in there in that enclosure for 3 weeks to a month, just depositing his DNA around the place. They seem to be quite happy and healthy so far. I've checked on them this morning after the heat that we had, and I found some alive snakes, so that's a huge bonus. You might be asking yourself, of all the reptiles in Western Australia, why venomous tiger snakes? And they actually are a great candidate, and they're going to be a bit of a poster child for us because they're prolific around Perth and the greater southwest. They're really easy to find them, surprisingly easy to catch, but they also occupy lots of different substrates. So they're not just on land or they're not just in water. They love the water, and they also are on the land. So we can trial lots of things when you're looking at what substrate we might be sampling. What's also important for another side of this project that we're really excited about, that's part of another grant that Morten has as well, Discovery Grant, but also part of this. From the enclosures, we know that tiger snakes have very genetically distinct populations from other work that's been done at Curtin University. So a tiger snake at Pilbara Lake is very genetically different from a tiger snake at Herdsman Lake or down south. We also know that there's whole genomes available. So what we want to also do is not only test substrates and test what we can sample, but also see if we can finally, from terrestrial samples, pull out some population level data. So can we get beyond? This is Damien Latouche. This is showing the genetic distance between some of these tiger snake populations. Can we actually get our tiger snakes to cluster with their actual population? And kind of for the first time try and show that can we, from environmental DNA samples in terrestrial systems, actually pull out some of that population level data, which I think is really exciting. May not be possible, but that's also an important kind of discovery to make as well. So thank you all very much.
Again, fantastic work, Josh. It's one keeps getting more exciting. So, not so the first one. Anyway, are there any questions for Josh?
Just because he bagged you out.
Yeah.
No, are you hoping or praying it doesn't rain while the tiger snakes are in the enclosure?
I've actually been watering the enclosures down when it gets over kind of 35 degrees. I think we know that rain has an impact on the degradation of DNA, likely because it increases the microbial activity in the enclosures, but I also think that's a natural thing in the environment. So it would suck because it would wash a lot of the DNA away and likely it kind of leaches through the soil, I think. But I think it's also natural. So I think it's okay if it does happen. Mm.
Thanks. Interesting to see how hard it was to detect the reptile DNA. Do you think it might be helpful to just use reptile-specific primers so that you're not actually sequencing any of those?
Yeah, for sure. But I don't actually think there's a good reptile-specific primer, like a general reptile primer yet, and that's what we'll be looking at designing.
That's something you're thinking about?
Yeah, for sure. Yeah. That'll be the first thing the PhD student does. Yeah.
What sort of time frame are you sampling after you take snakes out of the enclosure? You can do a long temporal study. I know from other people's work it can last in the environment for quite a while, right? I think it's
yeah. We're still umming and ahhing a little bit, but it won't be that normal thing where people sample it an hour, 2 hours, 3 hours, right? It'll probably be more realistic, like sample 1 week after, 2 weeks after, 3 weeks after, and do that for a period of time, possibly. No one's done for reptiles over 10 days in anything like that. So we'll go past 10 days. But everyone gets to 10 days and says we can still detect reptiles. That's useless. So keep on going.
Great. We'll come to a close. Thank you. Okay. We're on to our last theme now. So this one will be on the new technologies. So we've got two speakers in here. Professor Simon Jarman talking on measuring abundance with eDNA, the big topic.
and then followed by Alyssa Budd, Dr. Alyssa Budd, who will be talking on using environmental DNA and natural capital accounting.
Thanks, Shane. So yeah, I've got this project on developing ecosystem indicators from eDNA abundance. There are a lot of aspects to this project. So I'm just going to talk about one of them today that I've been working on lately because it's what's in my mind and I'm most interested in. So if you do any kind of ecological sampling with whatever method you choose, you get a distribution of species and incidence or abundance that's kind of like this. You get a few things that are very abundant, a lot of things that aren't very abundant. But this effect is really exacerbated with eDNA, partly because it's a very sensitive method, so you can detect things over a very broad range, but also because this long tail of low-level occurrence of things is full of other things that aren't real. So it's full of contaminants from other experiments, remnants of things that died some time ago, but you can still detect, so new ancient DNA, if you can say that, and chimeric sequences that are created during the PCR process, so different molecules recombine and give you something that looks like it's real, but it's not. So as a result of that, we all filter our eDNA meta-barcoding sets to try and remove things, and we tend to focus on the long tail. So we've been doing some work lately where we have some very highly replicated sampling sets, and this creates a new kind of abundance estimate, essentially. So frequency is kind of like a quasi-abundance thing. So what I've been working on lately is how we can use the frequency of things as well as abundance to work out better filtering methods for eDNA data sets. So this is a graph of a long tail distribution of things that this is fish data from Port Hedland from the study I mentioned earlier, where we have about 1,000 samples. Each dot on that graph is a species, and it's on the y-axis is its relative read abundance in the data set. On the x-axis is its relative proportion of occurrence in the data set. And on that scale, everything kind of clusters down in this bottom left-hand corner. So it's not actually that useful. So we need to focus in on that long tail. And I just wanted to put a reptile in here. So this is like we can focus in on the long tail of that beautiful chameleon, just keep Morten happy. And anyway, this is actually what it looks like. That's what yeah, exactly. So that's what it is on a linear scale. You put it on a log scale, and it really focuses in on the bottom left quadrant, which is what we're interested in. And some things come out from this view of it. So things that occur once in the data set are in this column here at different levels of abundance, twice in the data set here at different levels of abundance, but it's the same kind of thing. The rare and low abundance things are down here. They're the things we want to try and get rid of. The really frequent and hyperabundant things are up in that top corner, the top right-hand corner, which are the things we don't want to get rid of. And I'll just talk about what people currently do. If you're looking for things that you really want to detect, you don't do any filtering at all. So if you're looking for invasive marine species like this green swordtail, or really rare and charismatic things like snow leopards that are really hard to find, you don't filter at all. You include everything. They'll probably show up down here, but it does mean you need to investigate everything in your data set, check that it's not a chimera or a contaminant or whatever, so that you can detect those things. So that's one established sampling strategy. It's what we do for all the IMS testing currently. The other thing that people do, and this is the most common thing, is when you're doing community comparisons, you tend to cut things off by a proportion within each sample. So the standard way to do it is to throw out everything that's less than 0.01% present in each sample. And on this kind of graph that I'm looking at, basically it looks like this. You draw a horizontal line and you throw out everything that's in red. I have found one paper where people investigated this effect a little bit. They basically tried to find out how important it was to move that line up and down, and they ended up finding that it did make an effect, but they ended up just going with 0.01% anyway. So essentially what people do is that they just follow a standard method because other people have published on it. It's like people using the 97% sequence similarity that people did for bacteria back in 2000 and everyone's been doing since, because it just sounds like what other people did. So anyway, it is important because it does make a big difference. So to illustrate the example, this is another study where we looked at fish in Shark Bay from two adjacent communities. So Hamlin Pools hypersaline, this area is less saline. And because those communities are so close to each other, there's some swapping of eDNA at very low levels, or some larvae move between areas, or just a small number of individuals. If you don't filter the data set at all, everything starts to look more similar. So this is the fish in each area by abundance, represented by the bars at the top. If they're only present in one area, they get a colour, red for Eastern Gulf, blue for Hamlin Pool, grey if they've got a connection though. So if you don't filter the data set, that's the top picture, everything becomes connected, and the most abundant things in either environment are present in the other environments. If you filter it, you get more separation. So this is filtered down to 0.01%. The most abundant thing in Hamlin Pool then becomes unique to Hamlin Pool, and it would separate out in any kind of ecological analysis you wanted to do, if it was a Venn diagram or a multivariate analysis or whatever, things separate out more. So people do it for a good reason, but I think there are better ways to do it. Now that we do have some of these highly replicated data sets, we can actually use the frequency of occurrence as well as the abundance to start basically not drawing a horizontal line. It's sort of a simple idea, just drawing a curved line, more of a diagonal, that favors things that are frequent, because frequency is actually an indication that something is real. If things show up in your data set more often, then you probably should be looking at them. So basically it means you can kind of save this little triangle of species here and include it in your data analysis. So this is just an advantage of highly replicated sampling. Another thing you could do is actually just completely base your analysis on frequency. So you could throw out everything that only occurs once in the data set and just include things that are present multiple times. So we quite often do analyses where we run a very broad scale primer set, so 18S primers, just to find out what the overall community structure is of things that are forming the habitat. So we might want to find out whether it's a coral dominated community or a kelp dominated community or whether bryozoans are dominating. And the rare things don't really matter for that kind of analysis. So this kind of data treatment would be perfect for that. So essentially, and another advantage of that is that actually everything becomes completely sampled. So if you've ever done species accumulation curves with eDNA, the classic thing is that it never gets to the top. You never get a fully sampled curve. Everything keeps on going up forever, partly because you're just getting more and more rubbish as you sample more things, but also because it's a very sensitive method. If you throw out everything that occurs once, by definition, everything's become perfectly sampled, which is great. I mean, it's not just neat. It actually also makes real ecological sense. It means that all your comparisons will be among things that are fully sampled, which is satisfying. So to summarize, what people do currently is there are basically two filtering strategies available. You either filter not at all for maximum sensitivity and then just deal with the false positives. You do an abundance-based standard filtering method where you're probably throwing away some things that are pretty useful. I think we should probably, for most community comparisons, be doing a mixture of frequency abundance. And I'm working on ways that you can sort of define where that line should be from the data rather than just making it up once again. So it'll be some sort of combination of richness preserved versus probably species accumulation completeness or something like that. Or you could do frequency-based filtering as well if it's appropriate to the question. So as we get better sampling methods, more data in, we can come up with these improved methods for doing eDNA analysis. So that's my fairly simple point from this project. Thank you.
Great, thank you. That's put us ahead of time, actually. So has anyone got any questions for Simon? One?
Well, I've got one.
Great.
Do you have any idea as to the level of replication you think you might need? Is there some level of standardization that we might be moving towards?
I don't know. So that was across the whole data set. So that was, we had about 1,000 samples from that data set. So that makes it really easy. That's definitely way too much. I haven't actually yet cut it down. That will be part of what I analyze, actually working out what the minimum level of replication you would need to make that effective. But that's, yeah, it's a good question. It's a good thing to think about.
Yeah, very much.
I can say it and you can repeat it. I'll start it. Simon, I'm interested, do you think that is going to give you a rank abundance that you could actually use for looking at changes in time, changes over time?
Yeah, absolutely.
Because that will then suddenly mean you've got some sort of multivariate precision with the ability to detect changes over time. Whereas I think with the absence of present data, you would see that, and eDNA is really not delivering what it could.
Yeah, well, that's something we should work on then.
Just a quick one, microphones here. Is there any difference in the way you would treat taxonomic-free data as opposed to assigned species data when you use this approach? For any insights?
Yeah, well, that is a good question. So I guess actually for most of the taxonomy-free data, it probably isn't filtered very much, Angus. Yeah, so it's probably not.
Sorry, it is.
It is filtered, yeah. Okay, so yeah. So I don't know. It's a good question. It's probably something to think about. I would have thought a lot of the time, because you're looking for commonalities between things, that actually, yeah, you'd need some sort of level of filtering to make sure they weren't spurious. But how it would affect those sort of results, we'd need to experiment with. So yeah, it's a good thought.
But you're using mainly sequences that are common right across the science.
It's not about me. Don't bring me into it.
No, I want to bring you into it.
You wouldn't be filtering away rare sequences, certainly, in the context of the ticking.
Probably not, or probably not too rare. I mean, you'd want to do minimal things like exclude things that are only present once. You go for that 2 per sample minimum or something like that. But beyond that, yeah, I'm not really sure. We'd have to look at what effect it would have. Yeah.
Right.
Okay. One more. Yep.
I'm just wondering how you interpret the like wedge-shaped nature of that relationship.
Well, yeah, good question. So I think someone else has talked. So basically, yeah, that is interesting. So if there's a straight line between there, anything that falls on that line is about as frequent as it is abundant. There'll be some things up here that are more abundant than they're frequent. And there probably is a lot more interpretation to do with that. I mean, some things do fall on the other side of the line as well, in that so they're common but not that abundant. And that actually may be a useful thing you can analyze. It may actually bring out different functional groups from this kind of stuff as well, or different distributions in the environment. And that would be a good thing to analyze with covariates to try and work out what it actually means. Yeah.
Yeah, whether you can get it like patchiness and
exactly.
Other things. Yeah.
Exactly. Yeah. So it may actually have more value than just this is just a data processing tool that I'm coming up with, but there may be far more interesting ecological things that could come out of this view of it as well. Yeah.
Okay, great. We might draw that one to a close. So thank you for that. More discussions to be had over beers later, perhaps.
Okay, next presentation is Dr. Alyssa Budd talking on using a eDNA, the environmental DNA and natural capital accounting.
Too many acronyms. Yeah, thank you, Shane. My name's Alyssa, and yeah, I get to lead the eDGES project where we're looking at using eDNA in natural capital accounting, or NCA for short. So one of the big motivators behind NCA is that economics has a blind spot. So this is an image of some logging in a mountain ash forest on public lands in Victoria's Central Highlands. If we look at this from a traditional economic perspective like GDP, we get an increase in jobs, and we also get an increase in revenue from timber production. So that's all very positive. Traditionally, what we haven't been assessing in many cases is the value of this forest when it's alive and functioning. So this might include ecosystem services like water purification, flood prevention, climate regulation, habitat provision, and of course, biodiversity. And often what we find is that the value that we might generate from harvesting our natural asset is far less than the value that we would receive if we had maintained it in its functional or service providing state. But you don't have to take my word for it. So McCormick and Showers did a study on this published in 2019. And over a 1-year period, the Central Highlands region alone was responsible for $806 million in water provision to Greater Melbourne and Victoria, $1.2 billion in erosion prevention via soil retention, and $356 million in carbon sequestration. So these mountain ash trees are really good at storing carbon. And that compares to just $21 million in native timber production. So you don't have to be an accountant to start looking at these figures and be thinking maybe logging or harvesting this natural asset isn't the best thing I could be doing with it. But you wouldn't know that if you couldn't see those values in red there. So a great way to think about natural capital accounting is just putting nature on the balance sheet. I don't know about you, but I'd definitely be taking 21 million over nothing. So we really need to be seeing these numbers. One thing you might notice is missing from this account is biodiversity. So this is an image of a leadbeater's possum. It happens to live in these mountain ash forests almost exclusively. And it's the state faunal emblem of Victoria. So it does have quite a bit of value, but it wasn't able to be featured in these accounts. And we see this pretty consistently, that biodiversity is underrepresented in ecosystem or natural capital accounts. And there are a few reasons for this. One is that it's quite difficult to value. So we don't yet have biodiversity credits in the same way that we have carbon credits, although they are emerging. And another reason is that it's very difficult to see. So you can imagine if it was your job to find this tiny possum in this huge forest, it would be quite difficult to do and really very time consuming. And so that, of course, leads us to the opportunity, and we think that is eDNA. So eDNA is an indirect detection method for biodiversity. We don't need to see the plants or animals that we're looking for. We just need to be able to detect their DNA, which happens to be quite ubiquitous in most environments. So to illustrate this point, that biodiversity assessments from eDNA are really efficient, I'm going to use a couple of examples that you've heard about today. So Angus seems to have almost single-handedly sampled every single wetland in the southwest of Australia. Not quite, but it's quite incredible what he's been able to do. And Marina plans to do a similar thing to develop a terrestrial ecosystem health metric for the northwest. So it's kind of insane that we think these projects are possible in the confines of a moderately or even well-funded postdoc, let alone these brilliant people actually going out and making it happen. It's just wouldn't be possible on that sort of scale with traditional biodiversity assessments. So I want to talk a little bit about eDNA assessments for natural capital accounting. And to do that, I want to do a thought experiment with you guys. So we're going to use this poorly generated bit of land. It is AI, I apologize for that, as well as the UN System of Environmental Economic Accounting, Ecosystem Accounting Framework, more affectionately known as the SEA. So under the SEA, we would consider this whole thing our ecosystem accounting area. This is the bit of land that we as an organisation either own, control or steward. And we would split that into ecosystem assets based on the ecosystem types that we could see. So there's 3 ecosystem types here, and each of those would get a separate set of accounts generated for them. For the purposes of our thought experiment, we're just going to consider this whole thing as one ecosystem type, one ecosystem asset, and we're just going to build the one account. So the first thing we need to do is we need to value our asset here. And to do that, we're going to use the replacement cost method. So this is the actual value of ecosystem restoration. And I've taken this from the BNUT NCA pilot study. If you're interested in natural capital accounting, this is a really great example. But if we consider this whole thing used to be a mine site or a bit of agricultural land, and we've spent $1 million restoring it, absolute bargain, that's the value that we would give it. We could also apply a premium to that value based on its conditions. So if we pretend that this whole area is a wetland, we can apply Angus's aquatic to that area, and it's in the green, it's looking pretty good, so we might be able to apply a premium of 30%. If we didn't have a optimized ecosystem health measure, what we could use is Matt Campbell's cDNA that he's working on. So this might help us understand pre-industrial conditions, and we can use that as a baseline or reference to understand the condition of our ecosystem today. And that aligns really quite well with current expectations under the SEA. What we could do again is look for endangered species. So Josh mentioned before Ben Hayward's Pilbara olive python assay. So if we use eDNA detection methods like this to detect the presence of endangered species, we might be able to apply an additional premium to our value. And then the invasive species, so Simon spoke a little bit about Shirley's IMS or invasive marine species assay. We might use that to detect the presence of invasive species, but this time we'd be applying a discount instead of a premium. And of course, ecosystem service providing species. So for example, the groundwater species that Nicole is working on, my understanding is that they're very good at nutrient cycling and water purification. So we might be able to value those ecosystem services and include that value on our accounts as well. This comes with the caveat that we do need biomass estimates to do this. Hopefully the rank abundance work that Simon is doing in his eDGES project will get us one step closer to that. And it's also where the field of eDNA is moving more broadly. And my understanding is that we can already do this for the ecosystem services that bacterial species are providing. All right, so that's our account. So the total value of our ecosystem asset for 2024 is about $1.5 million. And if we are good land stewards and we do a better job at maintaining this natural capital, our ecosystem asset this year, we might have an increase in condition, we might increase the number of endangered species, we could decrease the number of invasive species, and it looks like our ecosystem service providing species are booming. And so as land stewards, we're pretty stoked that the value of that bit of land, because of how we've taken care of it, has increased. So you can see how these natural capital accounts can provide really good incentives for people who own, control, or steward land to take good care of it. So I'm running out of time, but I do want to talk about the project in general. So using eDNA in NCA, we've been thinking a lot about where eDNA can inform NCA or natural capital accounts. I'd really like to focus next on methods and metrics. I think eDNA has a real comparability issue that we need to address if we're going to use it for NCA purposes. And so this might be standardization of very specific eDNA methods, or looking at statistical harmonization techniques to be able to bring together metrics from different eDNA survey types and get meaningful and comparable data from them. From that, we might build a set of sampling recommendations. So in a lot of cases, eDNA surveys just don't have the replication that we need to apply some of these statistical methods that we'd like to do. And then finally, the valuation step. We have a couple of colleagues in the School of Accounting at Curtin who are really keen to sink their teeth into some of the biodiversity metrics that we generate from eDNA, and they can do that final valuation step and then build a set of accounts. And that's all from me. Thank you.
Fantastic, thank you, Alyssa. Okay, I'll throw it over to the audience if anyone's got any questions.
Shane, can I ask a question from me? When are companies supposed to be producing natural capital accounts?
Asking you.
Yeah, okay, so the question from Ewan was when are companies supposed to be producing these natural capital accounts? So I'm going to start with natural capital accounting is kind of like this umbrella term. What I've talked about here today is the actual set of accounts that we might make under frameworks similar to the SEER. And we don't know when that's coming, but there is this other arm of it that's more about risk assessment, opportunities and dependencies. So you don't necessarily have to own the land, but if your company happens to rely on natural assets, a good example is pollination. Then very soon, probably in the next 3 years, things like TNFD, so the Task Force of Nature-Related Financial Disclosures, they are going to become mandatory in Australia. So within the next 3 years, we're going to see this stuff start rolling out, and we're really going to have to be ready, hopefully, with some eDNA metrics that we can offer companies to be able to assess their natural assets.
I have a question from Andrew Herbert online. And the question is, are you thinking about this for the nature repair market too?
That's a great question. I'm not familiar with the nature repair market. Is anybody here? Well, I guess the short answer is no then. But if it's useful for it, please email me. I'd love to talk more about where these types of things that, can be implemented.
Thanks.
Yeah.
Right. Shane, can I just ask one?
Thank you for a good talk. It's good to see it like this. But so I take it that you say that of course eDNA can be beneficial in this context because you could produce a lot of data fast, right? I mean, that would always be benefit when trying to account for something like this or put a value on it. That's a benefit. But you also suggest that maybe we can come up with a new metric, something that we can implement into this NCA system. Is that correct?
Yeah, so potentially a new metric or an adapted metric. So something like aquatic or other chickies that exist and are being generated everywhere are really good because ecosystem health is part of this natural capital accounting framework. But I think in terms of metrics would be, and I'm thinking a lot about comparability. So if it's just something simple like species richness, we know that if we're not reaching the asymptote on our species accumulation curve, then richness at one side is not comparable to richness at the other. So something Simon and I have been talking about was, well, how do you get that to be comparable? And filtering might be one way. But yeah, thinking about metrics, I guess, more in depth than that and maybe a bit more complicated. Can we get beyond richness in a way that's comparable? You could have something that's nationally applicable, so then the valuation methods that are applied are going to be standardized. Whereas if you are applying valuation methods from individual surveys that are all a bit different, then they're not going to be directly comparable. So those are the types of things that we want to explore. Yeah. I hope that wasn't a politician's answer, by the way.
Great. All right. Thank you very much, Alyssa. Cheers.
Okay, so that brings us to the conclusion of the presentations today. Hello? Oh, one moment, we've got a question.
I was just wondering how all of the information that you're gathering, if you're thinking about how it might be used to better develop policy and strategy for conservation for WA and nationally, and if you're starting to think about mechanisms to input what you're learning into, you know, what we're doing in current systems and practices.
Tough question, but I think the last project you've seen here is a very good example of the takes on this basic fundamentals in research and development. So basically speaking, like was in there. I mean, to put it broadly, right? But we are almost all the people you've seen presented here today would probably characterize ourselves as conservation scientists, conservation mindset, and to us it's a little bit of a new field. So I should be playing this, but we also do realize that if we need to make a real change, we cannot only sit in our ivory towers and keep publishing papers. We need to come up with we need to learn how to speak that language, actually develop new tools. At this stage, I don't think I could be any more concrete than that. Maybe some of my colleagues can be more concrete, but I think this is one of those projects where we're really trying to make think differently, rather than just presenting a new biodiversity assessment of a given area. We're trying to actually come up with a tool that can feed directly into this natural capital accounting, so we can start speaking with politicians in a language they understand. Right? Anybody else have anything to add?
I'll add on co-lead on these projects with Morten, but I think one of the big things we're finding within the government agencies, they're getting more and more tight for funds, and monitoring programs are being cut, and yet we've got more and more pressures, particularly with things like climate change. One of the big things that actually starts to pull this data together is how we can actually monitor ecosystems. And for once, I think they're actually, through the combination of the tools that are being generated, going to be systems and metrics and data analyses for doing ecosystems-based monitoring that government agencies and ordinary populations can actually understand whether systems are improving. So there's the THEAT that's been developed in New Zealand as well, but also multivariate control charts that can actually take all the data and make sense of whether a system is improving or not improving. The challenge is then communicating that information, which is something that not so much eDGES, but the trend lab is focusing on, and it is.
Perhaps just to add to this discussion, I'll ask a question. Are all the data sets that are being generated through the eDGES program being made publicly available? So as a sequence data being uploaded to GBIF, for example, it can be leveraged by government agencies for conservation purposes and other things?
That's a great question, and yes, we are. And I'm going to throw over to our data scientist extraordinaire for a few comments.
Our data into a standardized format, put it in our own internal SQL database. We can query ourselves within Curtin, and then we're currently working on a bridge to essentially have an automatic upload once the data sets can be made publicly available. They can automatically be uploaded to a platform such as GBIF, and also compared automatically, right? So we want to also be able to pull data down from GBIF and make comparative analyses on our data before we make the data public. It's a verification process. So yes, we're early next year we'll be releasing a tool online where you'll be able to look at all of our data and in a similar way to GBIF, and then that's the information that sits behind that tool will be being passed directly into the GBIF.
That also include samples as well.
the sample data. Yeah. So that, I mean, making all of this data freely accessible and making it interoperable and following the FAIR principles, I guess, is making sure that the rest of the world can use this data. So we're not operating on an island. Great. Okay. I'm going to switch across to this one now. So yes, we'll draw this to a close. And so now I'd like to introduce Dr. Tim Cooper. Tim Cooper, who's the head of biodiversity and land in the environment group at BHP, to say a few words. So Tim has been absolutely instrumental in getting this going, and we've really appreciated just having a science chat with you. So we'll look forward to handing over to you now.
Thanks, Shane. Thank you very much. Yeah, look, thank you. I'm not sure what more I can add to that conversation that we've just had in indeed the last few hours, but you want me to press? Okay, so yeah, okay, thanks. Yeah, look, I think I make this joke every year, but once a year I'm reminded that I did do a PhD at some point in my life. Malnutrition for a couple of years, got down to about 80 kilograms in weight, survived, and here I am, a couple of decades later in this biodiversity role. But I thought I might reflect more. I was writing some notes down as we were going about how this project has impacted lives, and in actual fact, what it's done for me inside BHP. And it was interesting that you spoke about the eDGES lab, and I can't wait till it's like the Curtin University. Is that next? Who knows? But this project, as you alluded to, started about sort of 5-6 years ago. A conversation I was having with one of our leaders at the time was around how do I measure biodiversity and have those conversations inside a mining company. And so it started off, because I'm a marine person, it started off with invasive species. And I guess this is a summary of the project that we see today. I do just want to call out that I didn't make these slides, and I know for next year that I'll get a reptile put on the slides that are made for me. But when I looked at the run sheet this year, and I saw that I wasn't following Angus, because you were a hard act to follow last year, Angus. I was so I'll come to your defence. I know they bagged you out before, but it was I was actually pretty relieved that I wasn't talking after you. But I mean, to Alyssa, it's a great way of summarising sort of the thoughts that we've had through this project over the last couple of years to see you sort of putting into natural capital accounting framing and terminology is really great, because they're the sorts of questions that, as enviros in big companies like BHP, and there are other representatives in other companies in the room, we're asked to understand our impacts on the environment. How do we measure and quantify biodiversity? And then how do I then translate that into a language that people in my business, my company, understand? And I think I was listening to Mattia talk before about the Salada Punta Negra project. I was lucky to be in Chile a couple of weeks ago, and I was talking to the asset president about that project and talking to him about eDNA, the importance of supporting the work at Salada Punta Negra. And I was thinking, like five years ago, I probably wouldn't have been having or being able to have that conversation with someone at that level. Asset presidents are like pretty important people in our business. They make decisions, they look after safety, but they're all about operations. And so this project, the eDNA, whether you're talking about wetlands or the tiger snake Airbnbs or whatever it is, you've allowed me to go into our business and have these types of conversations around biodiversity and how we measure it. So yeah, thank you. Thank you for the work you do, the impact. To the question before about informing access to data and policy, this funding is not for the benefit of BHP. It comes out of our social investment framework, and the idea is that the data and the papers are published and made available. So this is not for us. It's for better management of the environment, not just in Australia, but globally. So yeah, next year we'll get a reptile put on my slide, although we've got one here, I guess, so that covers up for it. So yeah, this is just a little bit about our social investment framework, where this money comes from and what it's designed to do. There's more detail on our website about it. I'm not going to go through the detail now because I'm conscious that we're sort of on time. Shane, is that right? Pretty much. Okay. Look, happy to take any questions. Otherwise, welcome you to come and have a networking refreshment. Whereabouts, Shane? Is that
next slide? WC.
Next slide. Thank you for attending. And